Deoxyribonucleic acid is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth and reproduction of all known organisms and many viruses. DNA and ribonucleic acid are nucleic acids. Alongside proteins, lipids and complex carbohydrates, nucleic acids are one of the four major types of macromolecules that are essential for all known forms of life. The two DNA strands are known as polynucleotides as they are composed of simpler monomeric units called nucleotides. Each nucleotide is composed of one of four nitrogen-containing nucleobases with hydrogen bonds to make double-stranded DNA. The complementary nitrogenous bases are divided into two groups, pyrimidines and purines. In DNA, the pyrimidines are thymine and cytosine. The purines are adenine and guanine. Both strands of double-stranded DNA store the same biological information. This information is replicated as and when the two strands separate. A large part of DNA, for which RNA substitutes uracil, under the genetic code. These RNA strands specify the sequence of amino acids within proteins in a process called translation. Within eukaryotic cells, DNA is organized into long structures called chromosomes. Before typical cell division, these chromosomes are duplicated in the process of DNA replication, providing a complete set of chromosomes for each daughter cell. Eukaryotic organisms store most of their DNA inside the cell nucleus as nuclear DNA, and some in the mitochondria as mitochondrial DNA or in chloroplasts as chloroplast DNA. In contrast, prokaryotes store their DNA only in the cytoplasm, in circular chromosomes. Within eukaryotic chromosomes, chromatin proteins, such as histones, compact and organize DNA. These compacting structures guide the interactions between DNA and other proteins, helping control which parts of the DNA are transcribed. DNA is a long polymer made from repeating units called nucleotides, each of which is usually symbolized by a single letter, either A, T, C, or G. Chargaff's rules state that DNA from any species of any organism should have a 1:1 protein stoichiometry ratio, and one nucleotide unit measured 3.3 along. Although each individual nucleotide is very small, a DNA polymer can be very large and may contain hundreds of millions of nucleotides, such as in chromosome 1. Chromosome 1 is the largest human chromosome with approximately 220 million base pairs, and would be 85 mm long if straightened. DNA does not usually exist as a single strand, but instead as a pair of strands that are held tightly together. These two long strands coil around each other, in the shape of a double helix. The nucleotide contains both. A segment of the backbone of the molecule is called a polynucleotide. The backbone of the DNA strand is made from alternating phosphate and sugar groups. The sugar in DNA is 2-deoxyribose, which is a pentose carbons, the prime symbol being used to distinguish these carbon atoms from those of the base to which they. Deoxyribose forms a glycosidic bond. Therefore, any DNA strand normally has one end at which there is a phosphate group attached to the 5' carbon of a ribose to each DNA strand. In a nucleic acid double helix, the direction of the nucleotides in one strand is opposite to their direction in the other strand. The strands are antiparallel. The asymmetric ends of DNA strands are said to have a directionality of 5' end and 3' end, with the 5' end having a terminal phosphate group and the 3' end a terminal hydroxyl group. One major difference between DNA and RNA is the sugar, with the 2-deoxyribose in DNA being replaced by the alternative pentose sugar ribose in RNA. The DNA double helix is stabilized primarily by two forces: hydrogen bonds between nucleotides and base stacking interactions among aromatic nucleobases. The four bases found in DNA are adenine and thymine. These four bases are attached to the sugar phosphate to form the complete nucleotide. As shown for adenosine monophosphate, adenine pairs with thymine and guanine pairs with cytosine, forming AT and GC base pairs. The nucleobases are classified into two types: the purines, A and G, which are fused 5- and 6-membered heterocyclic compounds, and the pyrimidines, the 6-membered rings C and T. A fifth pyrimidine nucleobase, uracil, usually takes the place of thymine in RNA and differs from thymine by lacking a methyl group on its ring. In addition to RNA and DNA, many artificial nucleic acid analogues have been created to study the properties of nucleic acids, or for use in biotechnology. Modified bases occur in DNA. The first of these recognized was 5-methylcytosine, which was found in the genome of Mycobacterium tuberculosis in 1925. The reason for the presence of these non-canonical bases in bacterial viruses is to avoid the restriction enzymes present in bacteria. This enzyme system acts at Li